Hello, everyone, and welcome to another meeting for Everything ALS, our Master Talk series. We've got two very exciting guests today, so we're going to jump right into it. I'm going to introduce them with a little bio for each one. First, Dr. Richard Silverman is the Patrick G. Ryan Aon Professor in the Department of Chemistry at Northwestern University. He is the inventor of the blockbuster drug Lyrica, marketed by Pfizer. He has another drug licensed to Ovid Therapeutics in clinical trials for epilepsy and a drug from his own company, AKV9, for which the IND application will be filed in May for the treatment of ALS. He has published almost 400 research papers, written 5 books, and is an inventor on 130 issued patents. Recently, he was elected to the National Academy of Sciences. Joining Dr. Silverman tonight is Dr. Ozdinler. Dr. Ozdinler received training in the fields of molecular biology, genetics, biotechnology, cell biology, anatomy, and neuroscience. She is the recipient of Harvard NeuroDiscovery Center and NUCATS Translational Innovation Awards. She was recruited to be the founding director of the second Les Turner ALS Laboratory at Northwestern, which focused on the biology and pathology of upper motor neurons. She is now a faculty member at the Chemistry Life Sciences Processes Institute, Les Turner ALS Mesulam Center for Cognitive Neurology and Alzheimer's Disease, and Robert H. Lurie Comprehensive Cancer Research Centers. I welcome them both. Between the two of them, I'm sure we're going to learn a lot today. So Dr. Silverman and Dr. Ozdinler, welcome.
Okay, so I'm delighted to be here with Hyundai. And I'd like to tell you about a molecule that we're hoping to file our IND for in the next month or so. Let's see what happens in clinical trials with it. And so I'll lead off by telling you the story of how we got to the point of identifying this molecule and some of its earlier pharmacokinetic properties. And then Dr. Ozdinler will take over with all of the beautiful work she's done to show that it is effective in vivo for protection of protein aggregation in mice, and that it has great promise. So this whole thing let me move this out of the way. The panel, no, didn't do it. Hide floating controls. How about that? Okay, good. So this all got started because we are interested in trying to inhibit protein aggregation because that's really the hallmark for all neurodegenerative diseases. And in the collaborators I had at the time, we were very much interested in amyotrophic lateral sclerosis. And so that was the first one that we focused on. And there we go. So I don't think I have to tell this group about ALS since that's the main focus of the group, but it's a horrendous disease, and generally death occurs in 2 to 5 years after diagnosis. It's mostly a sporadic disease. About 10–15% is hereditary. And about a quarter of the familial disease occurs from mutation in the gene that encodes superoxide dismutase 1. The mutant form of superoxide dismutase aggregates, and that causes the neurodegeneration. There are only 3 FDA-approved drugs: Riluzole, Darvone, and this AMX0035. And so we need to get things that are really effective, since these to date have not been highly effective. So the reason mutant SOD1 can cause problems is because it seems to inhibit protein in the endoplasmic reticulum that is important for transport and degradation of the proteins that are marked for degradation, ubiquitinated proteins. And so that gets blocked. And so these ubiquitinated proteins aggregate and accumulate in the cytoplasm. And that's a hallmark for ALS. Both the mutant form and the wild type form interact with a protein that activates an enzyme that is important to our immune system. It produces superoxide to fight off pathogens. And wild type superoxide dismutase is a regulator of this process. However, the mutant form of SOD1 binds strongly to this activator of the enzyme that produces superoxide and sort of locks it into the active form. So you get a large production of superoxide that results in damage to the motor neurons, causing aggregation. So we thought we would do a high throughput screen. This is going to be a phenotypic screen. Since we don't know a target, we're just looking for molecules that inhibit protein aggregation. There we go. And so we set up two screens, one to look for aggregations, and then the other, the toxicity resulting from aggregation. And what we found early on is that we really couldn't see the aggregates, because as soon as they started to form, the proteasome would hydrolyze these aggregates. And so we needed to add an inhibitor of the proteasome, namely MG132. And by doing that, then we could see these aggregates and could see what happens when we added molecules to them. And so in conjunction with a small startup company, a high throughput screen was run, about 50,000 compounds. And initially we had 17 series of compounds that were identified as hits. We did a bunch of computational filtering of these molecules to see which ones really are similar, and then bought analogs of these and tested them to make sure that it wasn't just a single molecule, but a series of molecules that would have the activity. And then eventually, after looking at the different series and applying a lot of medicinal chemistry qualities to them, we honed it down to 3 series that were active in both those assays. Today, we're only going to talk about one of those series, the one that led to this molecule, NU-9. So this is the series, initially called cyclohexane diones, but it's actually in equilibrium with the enol form. And we saw a number of the molecules that had that structure, and that of the 50 that we had, 10 of them actually had activity. We found that if you modify this top ring, you lose activity, or if you cut off the bottom ring, you lose activity. And so we really needed to focus on this general structure like that. And the best of those molecules that we tested was this one with a fluorine para, and it had really all the properties we were interested in terms of solubility, metabolic stability, and permeability. The only thing it didn't have was potency. And so it was pretty weak. And we found the other molecules, the same thing. Everything looks good, except the potency was weak. And so that's what we were really focused on at the beginning. And so, of course, as a medicinal chemist, we're going to look at electronic effects, positional effects, steric effects, changing substituents around here to see how that would affect its potency. We found that you needed to have a substituent. If you just had the unsubstituted ring, it really didn't have the activity. So it was a matter of just finding out where these substituents should be on there. And so we carried out a halogen screen, putting halogen at different positions to see which was best. And it seemed the meta position was giving the best results with fluorine, same with chlorine, same with methyl. And so that was the position that we really started to focus on. Seemed that you could add a fair amount of bulk at that position. And eventually we found by putting 2 trifluoromethyl groups on there, we had at least sub-micromolar potency with this. So that molecule then was one we wanted to proceed further, try and develop. We said potency was okay. Solubility good, stability, permeability. It didn't have an effect on this potassium channel in the heart, the hERG channel. It can cause arrhythmias. We tested it against a number of different pharmacological targets, and it didn't interfere with these other pharmacological targets. So it seemed like it was pretty clean. So then we moved to animal studies with this, and we found that maximum tolerated dose, 75 mg per kg, that's pretty reasonable, and it had really good brain penetration. So we knew we would at least get it into the brain for activity. We were really disappointed, though, when we put it in the ALS mouse model. And so this is a standard model. It's a mouse with mutation in the gene that encodes for superoxide dismutase 1. And it was sort of surprising that it didn't have an effect on the survival, because at the time, that was the way you determined if a drug had potential, is if by treating this mouse with the molecule, the mouse lived longer. And so we're trying to figure out, well, why is that? Why didn't it have that effect, given that the 2 other classes of molecules that we were studying that didn't have properties any better than this really did have an effect. And so here's one of them, the one on the left here, did have activity in the ALS mouse model. The half-life for both of these molecules is about the same. CNS penetration was about the same for both of them. The one thing that seemed to be different was this volume of distribution. That it seemed the one that was active had a much larger volume of distribution than this cyclohexane-dione. And so maybe it got into the brain, but it just didn't get distributed to the motor neurons that we needed to see the activity. And so we took a look at this molecule in other cells, cell types, to see how it fared. And sure enough, the only cell type that it seemed to be active was PC12, which happened to be the cells that we did our initial high throughput screen. So that's the only reason we saw this, was because it's active in that cell line, but it's not active in any of these others that the molecule on the left was active in. And then furthermore, if you looked in cortical neurons, the molecule on the left was active in cortical neurons, but not our cyclohexane-dione. And so it seems that it does get into the brain, it just doesn't get into the neuron. And so that's why we're not seeing an effect in the mouse model. And so what we needed to do was to figure out, how can we get this to get into the neuron? And I had a remarkable postdoc at the time that had the hypothesis that maybe there's too much rigidity here, and that you need to open this up and give it flexibility so that it can maybe find its way to the neuron. So she added these linkers in between the 2 groups. And sure enough, both of these molecules were active in the cortical neuron assay, as was this one down here. This one turned out to have the better potency and PK. And so that was the molecule then we decided to continue with. And as I said, it is active in primary cortical neurons. The molecule has this chiral carbon, so there are two enantiomers. We made both enantiomers, and this S isomer is the one that has more potency than the other one. And both of them also are active in cortical neurons. And so this molecule was the one that we wanted to go further with. And so we looked at other properties, pharmacokinetic properties. Plasma stability was good, microsome stability, brain penetration was outstanding and quite orally bioavailable. Penetrated membranes well without efflux. It had almost no metabolism, and so that's great. You don't have a bunch of products that are being produced that could cause issues. And I said it had 94% oral bioavailability and pretty good kinetics, both in plasma and brain. And sure enough, this one does extend the life of the ALS mouse about 13%, which is comparable to what Riluzole did initially. And so we look at the other off-target effects. It doesn't seem to inhibit dramatically the cytochrome P450s that are important for metabolism, and it could cause drug-drug interactions if you did interfere with those. Said it doesn't interfere with the hERG channel in the heart that can cause arrhythmias. And so then we wanted to really see if it had a therapeutic effect. And I was sort of at a standstill at that point because this mouse model was the standard in the industry, but it seemed all the molecules that were effective in the mouse model didn't translate to humans. And so it was almost foolish to even try to extend the life of this mouse model because it didn't then have any effect on extending human life or making this disease any better. And that's when I met Dr. Ozdinler and started to talk about her research, which was focused on upper motor neurons, the cortical spinal motor neurons, where she found that they degenerated very early on. These upper motor neurons are pretty much the same in mice as in humans. And so that maybe if we can improve the health of these upper motor neurons in mice, it would translate to improving upper motor health in humans. So Hande, why don't you take it from there and tell us how you worked with this molecule.
Thank you so much, Rick. And I must tell you, it's been a pleasure and honor working with Dr. Silverman. And I think we all know that he's also the inventor of Lyrica, and he has an extensive knowledge about medicinal chemistry. And in my opinion, he's one of the best medicinal chemists in the world. So I think it was one of the best days in my life that we met at the ALS Association meeting in Chicago. Marilyn Blondell was organizing a meeting. And even though we were at Northwestern, we did not know each other at the time. And we met over the lunch table and said, oh, you're at Northwestern, I'm at Northwestern. And we were at the University of Chicago. And as he explained, he had these compounds and, at the time, he also had the, I think, ALS Association grants, a couple other grants to characterize these compounds, but we did not really know if they would have an impact on the health of neurons that are diseased in ALS. And as Dr. Silverman has said, the cortical component of ALS has been totally left out of the equation, out of investigation. We didn't really know if any of the compounds that go into clinical trials would also improve their health. And people actually didn't really care too much. And the idea that cortical neurons are going to degenerate anyways, and that the disease goes from periphery to the spinal and then to the cortex, really diminished their importance for any therapeutic interventions. But now we realize that the movement actually starts in the brain, and the cortical component of the motor neuron circuitry that degenerates in ALS patients is extremely important. And if we improve the health of the diseased upper motor neurons, that may have an impact to the spinal motor neurons, because the spinal motor neurons get their input also from the cortex. So it is the circuitry that degenerates, and we wanted to investigate whether these wonderfully developed and characterized compounds would have an impact on the diseased upper motor neurons. And that gives a very unique approach to ALS drug discovery, because we want to start, fixing the brain. So then the proof of concept work was that can we really improve the health of diseased upper motor neurons? So Rick, do you want me to share slides, or if I say next slide, would you be able to?
Yeah, next slide.
Yes, next slide, please. Thank you so much. So, cortex is very complex. It's very heterogeneous. There are thousands of different neurons and cells, and it's only, the upper motor neurons are important, but other neurons are important as well. And how do you know which one is an upper motor neuron? They all look the same. There are really no markers. So that was one of the reasons that people left the cortex alone, because you can't really see, you can't really study them. So the first thing we did was to make a reporter line in which upper motor neurons are fluorescent, so that when you look in the brain, you exactly know which ones are the upper motor neurons. So that allowed us to shift focus from mice to neurons. Because if you compare mice with human, of course, we are different species. Mice is a mouse, mouse is a mouse, human is a human. But if you look at the upper motor neurons in a mouse and upper motor neurons in a human, at a cellular level, there's very good translation. And our goal is to improve the health of the diseased neurons at a cellular level, because if we improve their health, we may improve their function, their connectivity, and that's important. That's why first we make this reporter line. And next slide, please. And you can imagine the next move is to cross this reporter line with many of the disease models that are characterized and generated and characterized based on the mutations that are detected in ALS patients. For example, initially it was SOD1 G93A, but of course now there are numerous TDP models and which have the mutations in ALS patients which have the TDP, alsin mutations, profilin mutations, spastin mutations for HSP patients, profilin mutations, whatever mutation is detected in ALS patients, and if mouse models are generated that recapitulates human pathology, you can cross them with this mouse to have a reporter line in which diseased upper motor neurons are fluorescent. And that is very important, because now you don't just study one mouse, you study numerous mouse models, and each mouse actually is developed to understand a specific disease pathology. So SOD1 and TDP, for example, are different, right? And in the patients with SOD1 mutation, you don't usually see TDP pathology. And the reason why patients develop the disease is very heterogeneous. And that's why we have numerous mouse models, because they all represent a subpopulation of the big ALS population. And the good thing is, when you dissociate the motor cortex from these neurons and put them in culture, you would exactly know which one is the upper motor neuron, because they become fluorescent. So among all cells, you would see the upper motor neuron. So that will begin to allow to perform drug discovery or drug verification studies, because you would look and see if the diseased upper motor neurons like it or not. Now, of course, this again brings the equation from mice to neuron at a cellular level, right? And if you think about it, patients develop the disease when their motor neuron circuitry fails. And the motor neuron circuitry fails when the neurons and the cells that make up the circuitry fails. So if we can improve the health of the neuron, then we can improve the integrity and function of the circuitry. And when we improve the function and integrity of the circuitry, then patients will be able to move their arms, will be able to swallow, and then we will have less and less of the pathology that are observed in the patients. Therefore, the core is the health of the neuron. So the next slide, please. Again, this is just to show that we can do in vitro studies and we can also do in vivo studies, that you can apply the compound of interest either by oral delivery with gavage or IP injection. You can put them in their water, drinking water, or you can also develop gene therapies, viral gene therapies, and then investigate whether these disease neurons in the brain would be resistant to degeneration, would remain healthy, would retain their cellular architecture, so you can bring cellular clarity to our analysis. Next slide, please. Okay, so this is exactly one of the experiments that we have done, in which in the culture, you can see these GFP neurons, right? They have long axon, beautiful. And they also have express markers that are specific to upper motor neurons like CTIP2. And the beauty is when the neuron, the upper motor neuron is healthy, they have longer axons and they branch and arborize. But if they are diseased, they have shorter axons and they don't branch and arborize. So this is very good because this becomes the outcome measure. This becomes a way that we can investigate whether the compounds of interest would have a beneficial impact on improving their health. Because now we have two outcome measures, axon length and the extent of branching and arborization. And these are quantitatively measured, and whoever measures it, it's always the same, that the diseased neuron have short axons and the diseased neurons have less branching and arborization. As you can imagine, in the next slide, we added NU-9 to the culture, right? And if NU-9 makes the neurons happy, then the neurons would respond by having longer axons, branching, arborization. So they show that they're happy. And yes, indeed, that's what they have shown, that they have now longer axons. They now have, and in comparison, we have also done these experiments in comparison to the two FDA-approved drugs at the time, Aderavone and Riluzole. So Aderavone and Riluzole, they did increase the the axon outgrowth, but not to the same extent as NU-9 did. So NU-9 was more effective than Riluzole and Aderavone. Next slide, please. And if you look at the combinatorial treatments, for example, if you add NU-9 together with Riluzole and NU-9 together with Aderavone, we did not know what we are going to get, right? But at the end of the experiment, we realized that there is an edit effect, which is very exciting and interesting because NU-9, I'm going to tell you more about the mechanism, but the mechanism of action for Aderavone is free radical scavenger, so it reduces the free radicals, and Riluzole actually basically acts on astrogliosis and microgliosis, so they are very distinct pathways. And when used together with NU-9, they don't block each other, they don't compete with each other, they help each other. So then now neurons have even longer axons. We take tiles. Sometimes to take the whole image, we need to make like 5-6 different tiles to take the whole image of the one neuron. So neurons were extremely happy. And this was shown in those images. And next slide, please. And they showed also extensive branching and arborization. Look at these, like with the addition of Aderavone, now they have even longer axons, they even branch and arborize, they're extremely happy. This is what we would like to see. We would like to see these diseased neurons becoming happier and healthier. And the diseased ones are usually short and stunned, and they don't really have arborizations. And now we can change their morphology, we can make them happy. Next, please. So this was very reassuring that NU-9 actually help improve their health in vitro in the culture. So the culture and in vivo may be two different stories, right? So then we decided to investigate whether giving NU-9 to live mice would also improve the health of the upper motor neurons. Next, please. Yes. So we have given them via gavage, and we started giving the NU-9 at P60 because that is about the time the mouse starts showing some symptoms. So it is very similar to a patient realizing there's something wrong with their body. And if a compound can begin to work or begin to be effective at that stage, I think it's pretty good because then we can target not only familial cases without symptoms, but also sporadic cases. So that's what we did. And initially we started with SOD1. And here you can see that upper motor neurons are in layer 5 of motor cortex. This is a very good reporter line. Not all neurons are GFP. It's just the upper motor neurons GFP in layer 5 of the motor cortex. This is very important. Then when you give NU-9 to SOD1, we realize the cells are still there. Because normally when there's no NU-9, in panel B, you see that there's very few left because they degenerate. They get cleared and you don't have very many upper motor neurons left. But with NU-9 treatment, we were able to retain them in the motor cortex. They did not degenerate, and to such a level that this 100 milligram per kilogram dose, if you count the cells, they were pretty much similar to the wild type CSMN numbers. I think this is remarkable because within 60 days, you know, when we know that there's ongoing degeneration, there's no degeneration, or, you know, I think at a cellular level, these cells, they decided not to degenerate. So we must be really making them happy to make that decision. So next slide, please. One of the phenotypes or one of the cellular pathologies that we observe in these upper motor neurons is that their apical dendrites, the extension of the cell that goes to the top of the cortex, they become degraded, they become filled with vacuoles, and that part is extremely important because that is the section where, or segment where other cortical neurons talk to them, or, you know, they make connections through their apical dendrites. And we realized that in the diseased upper motor neurons, these apical dendrites filled with vacuoles, they degrade, and the same thing is observed in the human upper motor neurons as well. But when you give NU-9, guess what? The apical dendrites did not degrade as before. There, we don't see that many vacuoles. The cytoarchitectural integrity was retained. So the cells not only retained their cell body, but also they retained the integrity of their apical dendrites. So this is actually very significant and important, I think. Next slide, please. And we counted those. We counted the apical dendrites that are healthy, and you can see the apical dendrites that are degraded, and the numbers were very much reduced almost to the wild type levels. So this is also very promising, and it's only 60-day treatment. Then we looked at the misfolded SOD1 levels, and yes, the misfolded SOD1 levels also showed a significant decrease. But imagine that this is only 60-day treatment, okay? So just like, you know, 2 months. And even within that time, we were able to see significant reduction. And I think that is very remarkable. Then we said, let's look very deep inside their cytoplasm. It's as if, like, you know, somebody's in the house and you peek through the curtains and, you know, you just want to see what's inside the house, right? So we can do this by electron microscopy because these neurons are GFP, and you can actually do an immuno coupled with EM, and you can look exactly what is going wrong inside the diseased upper motor neurons. So we realized that with the NU-9 treatment, there are 2 major things that are happening. One of them is that in disease cells, the mitochondria is usually disintegrated, the inner mitochondrial membrane is broken, mitochondria is affected, and the endoplasmic reticulum, which is important for the production of protein, is again, it becomes loose, or it becomes like a balloon, or it becomes segmented. So there's really a problem with the ER. So those 2 are major problems, mitochondrial problems and ER problems in diseased upper motor neurons. And we realized that when we give NU-9, the mitochondria begins to have cristae structures, the inner mitochondrial membrane is more intact, and ER becomes stuck, chack, like, you know, in line, perfect. You don't see those disintegration of ER. So it seemed to be doing very well with this helping ER and mitochondria. Next slide, please. We quantified those, and you can see this in our publication as well. And we have looked at many cells and thousands of mitochondria. It's not just one or two. We have seen in almost every cell that we have looked at that there was improved mitochondria and ER. Next, please.
This slide is for TDP-43.
Okay, so let me then tell you, in addition to the SOD1, right, we made actually a leap asking whether this compound would also have an impact on the TDP model. Because protein aggregation is a common problem, right? But the proteins that aggregate may be different. Like in the SOD1, you may see misfolded SOD1 toxicity, but in the TDP pathology, you see aggregation of TDP. So different proteins may aggregate, but protein aggregation is the common problem. So if NU-9 is effective in reducing protein aggregation, which was identified using the SOD1 system, would it and can it be effective within the context of TDP-43? That was the major question that we asked. And we also gave NU-9 to the TDP mouse. And guess what? We realized even though the underlying cause of the disease is different, even though different proteins accumulate in those two different disease models, you basically get very similar results, that the mitochondria is improved, ER is improved, and on the next slide, that the corticospinal motor neurons again retained in the cortex, and the apical dendrites do not show disintegration and degeneration. So even though the mechanisms are different, NU-9 has a similar impact. And I think this is very important because, as you know, we have seen in clinical trials that, there's a clinical trial and a subpopulation of patients get better, but, it's never statistically significant. But if NU-9 has an impact both on SOD and TDP, and those are the two non-overlapping pathologies, and they represent a very large population within the ALS population, then NU-9 may actually have broader implications to a larger ALS population. Next slide, please. All right, so then again, one more, yes. So we think that AKV9, now it's AKV9, because Dr. Silverman actually formed a company with his sons and licensed the NU-9 to the company. And the company now applies to FDA to get approval for clinical trials to start, and NU-9 became AKV9. But for research purposes, we sometimes call it NU-9 as well. So now AKV9 acts on multiple cellular pathways. And those are the pathways that are common or commonly observed in other neurodegenerative diseases as well, such as protein aggregation, such as mitochondrial problems, such as ER problems, such as axonal transport defects, such as cellular integrity, like, loss of cellular integrity. And next slide, please. So when we give AKV9, as far as I know, correct me if I'm wrong, this is the first compound that can act on these 4 major or 5 major cellular defects that are in part responsible for neurodegeneration. Protein aggregation is reduced. Mitochondria regains its structure and strength. ER also becomes healthier and more intact. We don't see those apical dendrite degeneration, and the axon outgrowth and branching arborization seems to be restored. That's why AKV9, I think, has important impacts. And of course, we looked at whether that would translate to any improvements in function. But people working with mice would appreciate the fact that looking at the upper motor neuron and translating it to behavior is very hard in mice. And but again, we have seen that there was really no body weight change. And next slide, please. This was very important because the latency to fall from an upside down grid Okay, so this was a grid assay. And our lab and other labs also realized that this grid assay may have an implication for the involvement of upper motor neurons in the motor neuron circuitry, because you put them on a grid, you turn them upside down. They need to hold the grid, okay, and they're going to carry their body weight. So this hand movement and the strength in the hand is extremely important. And that's also one of the indications for the involvement of upper motor neurons, and seemed to be that the NU-9 treated mice did actually really well. And that's a good news for us. And next slide. And maybe Rick, you can take over for the orphan drug designation and how the clinical trials are moving forward.
Right. So since ALS is an orphan disease, less than 200,000 people in the US, we applied for and were given designation for this compound. And so, as Hande said, we started a company, AKV Therapeutics. We have a great group of people that are heading this. And our scientific advisory board are some of the top people in the world, clinicians working in ALS. The small scale synthesis that we worked out in my lab was translated to the large scale synthesis. So we have plenty of material for clinical trials at this point. We had done MTD studies in mice and went up to 1,200 mg per kg, and there were no adverse effects. When we then did a 7-day repeat, give the drug every day for 7 days, even at 300, there were no major effects. So the no observed adverse effect level is something greater than 300 mg per kg. So we started from a phenotypic screen with the idea of, let's see if we can find something that inhibits protein aggregation resulting from a mutation in the gene that causes ALS. And from that, we eventually evolved a molecule after many years of modification that not only maintains the health of upper motor neurons in mice that have a mutation in SOD1, but also in TDP43, which is a mutation that occurs in, well, 97% of ALS patients. And so we think this could be a paradigm shift in how you test drugs now for ALS and maybe other neurodegenerative diseases. So it took a lot of people to do this, and be delighted to answer any questions. I see there are a whole bunch in the chat at this point.
Thank you for that great presentation. We do have a few questions, so Brian and I will take turns going over them. The first one is, do these drugs only work for SOD1 ALS?
No, we've all the studies we've done in SOD1, we've done in TDP43, and we get as good or better results with that.
Yes. And currently, we are also testing them in the spastin mouse and in the alsin in the mouse and in the profilin, so different mouse models as well. So we will share those results, but I can tell you that they are pretty much promising at this time. And I don't think it's just one model. It's basically diseased upper motor neurons. And as long as diseased upper motor neurons degenerate due to these 5 mechanisms, right? I think that they would benefit from NU-9 treatment.
Okay, I guess the next question is, so what is the differences in mode of action in regards to the other 3 drugs for ALS? Yeah,
so as you know, riluzole is for reducing astrogliosis, right? And it is believed to act more on astrocytes than on motor neurons, because towards the end of the disease, especially, and sometimes, you know, during the disease, the astrogliosis has a huge impact on neuronal degeneration, because neurons and astrocytes are like husbands and wives, you know, they live together and they support each other. But during disease, something happens that they stop supporting each other and they start to fight. And when they fight, the astrocyte wins the game, and the astrocyte chews up the motor neuron, and astrocyte basically clears the motor neuron. And the riluzole is acting on the astrocyte, saying, "Don't do it. He's a good guy. Don't do it." You know, and that's how the first ALS drug works. The other one is a free radical scavenger. So there are many free radicals in the cell, and that may emerge due to many mechanisms, and one of them is related to mitochondrial dysfunction, disintegration as well. And then this free radical, you know, it's just like a binds and bumps and many other proteins. It creates a kink or damages the membranes, damages the protein. So it creates a like damaging toxic environment, and that is one of the reasons. And another one works on that. And NU-9 does not do this free radical or astrocyte. It actually improves mitochondria, reduces protein aggregation, improves cytoarchitectural dynamics, improves axon outgrowth. So it touches on different mechanisms. And I think that was one of the reasons why we have seen an additive effect, especially within the context of SOD1.
Thank you. Our next question is, does this help LMNs as well?
Do you want me to take this one as well?
Yeah, go ahead. Lower motor neurons. Yeah, sure.
Okay, so we studied lower motor neurons in these two mouse models, SOD1 model and the TDP model. So in the TDP model that we used, A315T, the spinal motor neurons do not undergo cellular degeneration to the same level and extent compared to the SOD1, which means even at the end stage, the number of cell bodies that are present in the ventral horn of the spinal cord is comparable to the wild type cases. That's why you cannot really do a quantitative study to investigate whether NU-9 treatment improves the health or not, because the cells are pretty much there, right? So we couldn't study the spinal motor neuron loss within the context of TDP currently in this mouse model. In the SOD1 mouse model, there is a very prominent spinal motor neuron loss. You begin to see spinal motor neurons as early as P60 and P120, significant reduction. So when we looked at the spinal cord of these mouse treated with NU-9, we did not see a prominent retention of the spinal motor neurons as we have seen in the cortex. That's why when we reported that NU-9 is the first compound to improve the health of the upper motor neurons, because it is our expectation maybe that motor neurons living in the cortex or motor neurons living in the spinal cord are basically the same, but some in the, live in the upper tower, or the other one lives in the, bottom part of the same apartment, but they are the same. But in fact, this may not be true, because upper motor neurons and spinal motor neurons are born from different progenitors. They develop differently. They have different transcription factors. They are distinct neuron populations. So, maybe one should not expect that one compound is going to cure them both. They may have different requirements, and we already know that they have different requirements for survival. But in the SOD1 case, we did not see, especially in the SOD1 mouse, we did not see improvement of the spinal motor neurons in the SOD1. But how translational is the spinal motor neurons of the SOD1 mouse is also questionable. So that's why we are actually doing iPSC cells now, from human different spinal motor neurons in the dish that are differentiated to spinal motor neurons in the dish from human cells. So hopefully we will have those results soon. And that would, I think, will be more informative than the spinal motor neurons. We don't know. So, but especially within the context of SOD1, the upper motor neuron was more prominent than the spinal motor neuron.
All right, perfect. Next question is, will individuals with ALS be able to talk again after receiving treatment with this drug?
Do you want me to take this again, Rick?
Go ahead. Yeah, if you have a crystal ball, go ahead.
Yeah, so I don't have a crystal ball, and it's hard to comment on this. And I don't know if they will be able to talk again. But I think if you think about the ALS pathology and why people develop the disease, it's because their upper motor neurons are degenerating. And if we can make these upper motor neurons happy again, so then they decide there's no reason for them to degenerate again. And if we start to treat patients at an early stage, such that the upper motor neurons are not cleared, they are still in the brain and they are responsive to treatment. In that case, I would imagine that the neurons would stay within the circuitry, and the circuitry may become functional, and there may be improvement in their outcome. And does that mean that they begin to speak? I don't know. But I would imagine that if we can make the neurons happy as early as possible, so that they don't degenerate, so that we can have functional circuitry, there may be improvements in their behavior.
What happens if you stimulate BDNF with these molecules?
I don't know. BDNF is brain-derived neurotrophic factor. It is a growth factor. And it is one of the growth factors that improve the health of motor neurons. And I have done studies with BDNF before when I was a postdoc in Jeff Macklis's lab in Boston. And we know that BDNF actually improves the health of upper motor neurons. But I have looked at axon outgrowth, branching, arborization with BDNF. And I think NU-9 was more effective than BDNF. And now, for example, the neuron study, even though it is called stem cell therapy, they're actually giving growth factors, and BDNF is included in their growth factor list. So I think BDNF is important, but having a small molecule treatment would be more effective than BDNF, because BDNF comes as pro-BDNF. You have to cleave it, and BDNF is not very stable as a protein. So there are many complications with growth factor treatment strategies.
Perfect. Next question is, do you guys have any updates on when clinical human trials will begin?
Okay, Rick, this is one is for you.
Yeah, so right now we're on schedule to file our IND as sort of mid-May. And then after that, it's going to depend upon, the FDA and raising appropriate funds to get it going in phase 1. Phase 1 clinical trials are expected to be with healthy volunteers, though.
No, I don't have anything to say.
Okay. How does NU-9 impact SPG4 patients?
So that's a very good question, and that's what we're trying to investigate, because SPG4 is a mutation detected in hereditary spastic paraplegia patients, and this is a disease where upper motor neurons are predominantly affected. And for the SPG4 patients, HSP patients, spastin mouse model is one of the best mouse model generated by Dr. Buss, and we are lucky to collaborate with them, and we have the mouse model in our lab. And we would like to investigate whether the upper motor neurons also degenerate due to similar underlying causes, meaning mitochondrial dysfunction, ER, protein aggregation, cytokines, all these. And if that is the case, some of them are common, like mitochondrial problems are common, but the axon transport or retrograde transport, the axonal component may be more important for those. I would imagine that NU-9 may have an impact on those as well. And that's why we're actually doing the in vitro and in vivo studies. We received grants from Cure SPG4 Foundation and Spastic Paraplegia Foundation. So hopefully there may be a utility for NU-9 for the HSP patients as well. But of course, this is experimental now. and clinical trials need to be performed. But first, we need to have a better understanding at a cellular and at a molecular level.
Perfect. The next question is, were the effects of NU-9 on reactive astrocytes studied?
We haven't studied active astrocytes within the context of ALS, but Rick, I don't know, have you studied astrocytes or?
Related to Alzheimer's, we've been doing a study in mice, and after 60-day treatment, we looked in the brains of the mouse, and astrocytes were healthy.
What about the neuromuscular junction? Is that impacted by NU-9?
We didn't study neuromuscular junction in detail because we have not seen a good impact on the spinal motor neurons, and we focused our attention to the upper motor neurons. I mean, we still have the tibialis muscle still in the fridge, but we haven't investigated the integrity of neuromuscular junction. So I don't exactly know the answer to that.
All right, perfect. This is gonna be the last question of Q&A, but will the neurons regenerate, or does this only apply to cells that have survived?
Shall I take the first step and then you? Okay. So this is actually a very important question, because do we really regenerate, or do we retain the health of the the degenerating neurons? So we begin to treat them at P60. And at that time, if you look at the electron microscopy studies, the mitochondria actually is very affected. And mitochondrial defects, we have shown that especially within the context of TTP, starts as early as P15, okay? So they have some mitochondrial problem that started much, much earlier. And then we even show that there are protein aggregations as early as P3, which means postnatal day 3 in the mouse, like they're this big, okay? But even when they're this big, there are protein aggregations beginning to occur in their upper motor neurons. So what we see as a problem is actually an ongoing problem that initiated much, 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 much earlier, right? And the cells actually can tolerate, and then when they cannot tolerate, they become vulnerable. And then they go like, I can't do this, And then after a while, they say, I can't do this. And they say, you know what, I'm done. Like, and there's a point of no return. So when we begin to give them the compounds before the point of no return, but when they're shouting and saying, I can't really do this, I'm stressed, right? So when we give it at that time, and we reduce their protein aggregation, we make their mitochondria intact, then they say, oh, thank God, thank God for cleaning my house. Thank God for giving me the energy, for allowing me to make the protein, for repairing my walls. And I'm okay now, I'm fine now. And I think at that time, the ongoing degeneration, the decision to, continue to degenerate is halted. And then they say, why do I degenerate? Like every problem is solved. And I think because you fix the mitochondria, you fix the ER and everything, the cells that were beginning to become vulnerable, now they're not vulnerable. So they may say, why do I become vulnerable? I can make ATP, I am okay. So I think it may work both ways, that the cells that were about to make this decision of no return, we may say there's no reason for you to take that route. And the cells which are going to become even more unhappy, then we take that back and say, there's no reason for you to be unhappy. And I think this treatment will help both ways. I don't know if I was able to explain. Rick, would you like to add something more, or?
I thought that was very dramatic. I haven't spoken to an upper motor neuron like you have.
Oh, I have been talking to them for a very long time.
Thank you, Dr. Silverman and Ozdinler, for your presentation and for answering all the questions. We ended up getting a lot of questions and some of them were not answered. Do you think if we email them to you, would you be able to take a look and answer them when you have some time, and we'll send it back out to everyone?
Sure.
That's
Thank
fine.
you.
And I it's 7:03 for me now, but I can stay another half an hour or so and I can answer more questions if you want.
I gotta get off now. So, but if you want to send out the emails with, I can answer what Dr. Ozdinler doesn't.
Okay.
Thank
Okay.
you.
All right.
Dr.
I can-
Silverman,
Thank you
thank you
so
so
much.
very much for
All right.
staying with us, and please join us again.
Sure.
Thank you.
All right.
All right.
Bye-bye.
And right
Yeah,
now,
rather than me answering emails, so ask me the questions while we are all here, because I have 7,000 unread emails in my inbox. I may not be able to answer them. So shoot, tell me now.
Brian, you want to go first?
Oh, should we answer the questions first, or should we move on to the open
No.
forum?
No,
Look,
we're going to
question-
answer the questions first.
Oh, we have approximately, give or take, 30 questions.
Okay, shoot, I'll be quick.
Okay. Okay, I guess to continue, so, okay, what could be the most important challenges to translate mouse treatment results to human treatment?
Yeah, so that's very challenging because what is the translation? Where does the translation occur is very important. So we realize, for example, mice is a mice and human is a human. So we can't really compare mouse survival with human survival. But the neurons at a cellular level, neurons in well-characterized mouse models and neurons in humans at a cellular level, they share a lot in common. And we published this in Acta Neuropathologica in Nature's Cell Reports, that, for example, mitochondrial dysfunction, ER stress, apical dendrite degeneration, whatever you see in the upper motor neurons in the mouse, you exactly see the same thing in the upper motor neurons of the human patients. So then I think the translation is at the cellular level. So if we learn to improve the health of the cell, the health of the neuron, that will be translational. Okay, next question.
Is there any realistic date for commercialization of the drug if everything goes according to plan?
Yeah, I wish, why don't you ask this to Silverman? But we are putting our IND application to FDA now. So hopefully our goal is to start phase 1 now. And we are we're rushing to complete the biomarker studies so that when it is time for phase 2 and phase 3, we will have biomarkers, especially pharmacokinetic biomarkers, to investigate the timing and extent of upper motor neuron loss. So, we want to move as fast as possible. And now we're talking to venture capitalists, we're talking to everyone, and our goal is to move this as fast as possible ourselves. So next question.
Could this drug be tested if one has PLS?
That was our goal, but unfortunately, when you do clinical trials, you have to select a disease. Like, you cannot say, I'm doing a clinical trial for ALS. Oh, let me also include PLS patients. So you have to have a clinical trial for a disease, right? And if that's the case, maybe either there may be a clinical trial for another disease, or maybe there may be a basket approach so that you have different diseases. So it's the scientific advisory board that we are having those discussions. But first, we must get the results from phase 1 that this is not toxic. And I think this is a debate, and this is an ongoing debate. And it's also the results from our spastin mice are important, because if we show that in the mouse, in the upper motor neurons, in the HSP model, it's not working, then it may not be feasible to include them.
What percentage of the ALS patients can benefit from this treatment? What do you think about this if we consider the different mechanisms causing the degeneration of UMNs?
I think, to be honest with you, I think a large population of ALS patients are going to benefit from this, because especially within the context of ALS, if there's really no upper motor neuron loss, patients are not diagnosed as ALS, because ALS is the disease of the upper and lower motor neurons. And that's why I think, ALS patients are going to benefit from this. And the mitochondria problem, ER problem, cytoarchitecture problem. So these are problems that are observed, in degenerating upper motor neurons. So I would imagine that it would have a broad spectrum impact on a broad spectrum. Okay, next.
Perfect. Were neurofilaments compared in the mouse models?
So neurofilaments can be used as a biomarker, and I don't know exactly the answer to your question because we have not done any biomarker study in the mouse. So we're not doing biomarker studies in the mouse. Our biomarker studies are done in human serum and plasma.
Did you observe Budina bodies in motor neurons after the treatment?
We haven't looked.
What is the criteria for healthy volunteers? And also, what is the age of onset for ALS? Is it within 2 years of symptom onset, or is it plus or minus?
I don't know. I think if we have any clinicians in the audience, maybe we can ask them. I don't know, but I think that ALS, I have seen some Alzheimer's patients, they were 2 years old and 3 years old. We published a paper studying their serum and plasma. So there are juvenile ALS cases, and there are some ALS patients who are 60, 70-year-old. I don't think that there's you have ALS within that age range. Like, I think it's pretty broad, but I am not a clinician. I don't know.
And then this is our last question before we hand it back to McFinn, but do antiviral treatments work, and specifically Aclovir as an antiviral drug?
I don't know what you mean by antiviral treatments. I don't understand.
Do we know who asked that question? Let's see.
Antiviral treatment, resisting the virus, or we actually use viruses for a way.
Yeah, I asked a question, Candace. Acyclovir, it's recommended by German scientists for an antiviral treatment to help ALS patients, and we do not use it in the US to date. And you have in your studies, you use viral in there, and I wondered if antiviral treatments was anything you looked at.
Okay, so I understand the question a bit better now. And the answer is no, we have not used them within that context, but we use viruses as a tool for gene delivery. So we are using adeno-associated viruses for gene therapy to upper motor neurons. But within the context of your question, no, we have not looked.
Thank you.
You're welcome.
Well, folks, another fine evening at ALS Talk Series. Hande, you are an angel. Thank you so much for saying that you had 700 emails in your box and that you'd like to just stay and answer these questions. Folks, that's how cool these speakers are. They're ours. We're a little possessive, dear. So anyway, every time you come on, you bring a lot of smiles to everybody because of your personality. So thank you so much from each and every person here tonight. I'm sure that everybody has gained a little bit of insight. There it is. It's the heart from Teresa sending it to Hande. That's it. That's it. Thank you so very much. I hope your evening is peaceful and your dreams are exciting.
Thank you so much for inviting us. And it's always exciting. And I am very optimistic and hopeful about NU-9 because I know that these are common problems, cellular problems that we see in numerous patients. And if we can make these upper motor neurons, the motor neurons happy, I think that's going to eventually lead to improvement of the health of the patients, because patients become diseased due to the lack of health of their motor neurons and of the motor neuron circuitry. So And I see actually Stephen has.
Yes, Stephen has, I was going to ask you to, Stephen, come aboard. Take yourself off mute. There you go.
Yes, sir. Can you hear me now?
Yes.
Yes.
How you doing, Hyundai?
I'm all right, thank you.
Yes, ma'am. Will you be administering NU-9 to the TDP 43 ALS patients
So,
in clinical trials?
so we will ask our scientific advisory board, and I think that some patients will also be involved in the, in those discussions. And we have not determined the inclusion criteria yet. So I don't know exactly what, but now that we have seen NU-9 actually, or AKV9 works within the context of PDB pathology, I would imagine that we should, or we would include patients with PDB pathology. But again, I'm not a clinician. I don't design inclusion criteria. So I don't know, but I would imagine yes.
Okay.
Thank you, Stephen, for asking.
Before we
And
leave, I just want to make sure that we answer all your questions.
Thank you.
does anybody else before we have our
Yeah.
sweet doctor go?
Yeah, I have one question.
Johnny.
On the clinical trials, he said that it was going to be looking for healthy volunteers. Does that limit your qualifications, your requirements for the program to people who are within 2 years of symptom onset? Or like me, I'm fairly healthy, I'm walking and talking, I can use both my hands and all. Would I be eligible at 7 years since symptom onset?
Okay, so in clinical trials, there are different phases. Phase 1 looks for toxicity, because even though we do toxicity studies in mouse models, in rat, dogs, cats, different mammals, but they want to know if this compound is toxic or not. Because if it is toxic, it's no go. You cannot do phase 2, phase 3. First, you have to confirm that this is not toxic. And that's done in phase 1. And it's a small group of volunteers, healthy volunteers. So you cannot use patients inclusion in phase 1. So that's why Dr. Silverman was saying that phase 1 will be healthy volunteers, because we have to pass the first phase 1, show that this is not toxic. And then in phase 2, now that we know it's not toxic, then you may include patients to the study. So, and I think that's the difference between phase 1 and other phases, because in the phase 1, first people look at toxicity.
Thank you.
All right, folks. Thank you, Handi, so much.
All right, have a good night. Thank you so much. And
Okay.
thank you so much for all the support that you have shown to our work. Please keep supporting us, because we are on a mission and time is the essence, and your support is very important. Thank you.
Know that better than anybody. Thank you.
Thank you.
Thank you, Candace. Okay. Casey, you can turn off the recorder.